Welcome to this video on the pituitary gland, the central endocrine regulator. We will explore its detailed microscopic anatomy, cellular populations, and related pathologies like gigantism and diabetes insipidus. The endocrine system is a network of ductless glands. The pituitary gland acts as the master regulator, operating under hypothalamic influence and maintaining homeostasis via tropic hormones. The pituitary gland is a pea-sized organ housed within the sella turcica of the sphenoid bone. It connects to the hypothalamus through the infundibulum and is divided into the glandular anterior pituitary and the neural posterior pituitary. The pituitary gland has a dual embryological origin. The Adenohypophysis, or anterior pituitary, develops from Rathke's pouch, an upward invagination of oral ectoderm. The Neurohypophysis, or posterior pituitary, originates from neuroectoderm. The Adenohypophysis, about 80% of the gland, includes the hormone-secreting pars distalis, the dividing pars intermedia, and the infundibulum surrounding pars tuberalis. The neurohypophysis consists mainly of the pars nervosa and the infundibulum, which is the neural stalk. The hypothalamo-hypophyseal portal system is vital for communication. It involves the primary plexus, portal veins, and secondary plexus, ensuring efficient transport of releasing and inhibiting factors to the anterior pituitary. Hypothalamic factors regulate the anterior pituitary's hormone secretion. These factors travel via the portal system, stimulating or inhibiting pituitary cell populations to release hormones that target distant organs. Histologically, the anterior pituitary, or pars distalis, features glandular cells in irregular clusters called cell cords. These cell cords are intimately surrounded by sinusoidal capillaries for efficient hormone release. Acidophils are chromophil cells with a vibrant pink or red eosinophilic cytoplasm. They include somatotrophs, which produce GH, and lactotrophs, which produce prolactin. They are generally smaller and more numerous than basophils. Basophils stain purple or dark blue due to their affinity for basic dyes. They constitute about 10% of the anterior pituitary population and include corticotrophs, thyrotrophs, and gonadotrophs. Chromophobes are non-staining cells that make up about 50% of the pars distalis. They can be either degranulated chromophils or undifferentiated reserve stem cells, appearing as, quote, naked nuclei due to their scant cytoplasm. The pars distalis is the main area for tropic hormone production, organized in cords of acidophils, basophils, and chromophobes. It is highly vascularized by fenestrated sinusoidal capillaries, optimizing hormone exchange. The pars intermedia contains colloid-filled follicles and secretes melanocyte-stimulating hormone. The pars tuberalis is a vascular sleeve rich in melatonin receptors, potentially regulating seasonal rhythms. The neurohypophysis is neural tissue composed of unmyelinated axons and glial cells called pituocytes. Unlike the pars distalis, it lacks epithelial cords and relies on a network of fenestrated capillaries for hormone release. Key features of the neurohypophysis include unmyelinated axons and pituocytes. Herring bodies are axonal dilations that store ADH and oxytocin granules bound to neurophysins before their release into capillaries. The anterior pituitary secretes 6 main hormones. These include GH, prolactin, ACTH, TSH, FSH, and LH, each produced by specific cell types like somatotrophs, lactotrophs, corticotrophs, thyrotrophs, and gonadotrophs. Growth hormone acts directly on muscle and adipose tissue to influence metabolism. Its primary growth effects are indirect, stimulating the liver to produce IGF-1, which drives bone and cartilage growth. Prolactin promotes alveolar development and maintains lactation postpartum. FSH and LH regulate the gonads through complex feedback loops, supporting ovulation in females and spermatogenesis in males. ACTH stimulates cortisol release from the adrenal cortex, governing glucose metabolism and stress response. TSH regulates thyroid hormone production, which in turn controls the basal metabolic rate. The posterior pituitary releases ADH, or vasopressin, critical for water homeostasis by increasing aquaporin insertion in the kidney. Oxytocin stimulates uterine contractions during parturition and milk ejection during lactation. Negative feedback loops are essential for maintaining hormonal balance. High levels of target organ hormones, like T3, T4, cortisol, and sex steroids, inhibit the release of their respective hypothalamic and pituitary tropic hormones. This scene is intentionally blank. Pituitary pathologies include hyperprolactinemia, often causing reproductive dysfunction. Somatotroph adenomas lead to gigantism or acromegaly. ADH deficiency results in diabetes insipidus, causing renal water conservation failure. Histological identification of the pituitary regions is key to understanding its function. The anterior pituitary is cellular with acidophils and basophils, while the posterior pituitary is fibrous, containing unmyelinated axons and herring bodies. In summary, the pituitary gland's dual origin results in specialized functions: the anterior pituitary's hormonal secretions via the portal system, and the posterior pituitary's neurohormone release via axonal projections. Understanding these axes is vital for endocrine diagnosis.